Test 1 has been graded. The scores are in the triple E box. The distributions and the statistics, such as the mean, will be visible sometime this afternoon along with the answer keys. If there is some issue, like a 0 is showing up and you're sure you were there or whatever, or if the number doesn't seem to match your estimate of what you would expect on the test, the images of the scantrons are being processed. They should be available in your Dropboxes on Friday, hopefully Friday at the very latest next Monday, unless there's some kind of problem. But this will be an image of your scantron so that you can look at it side by side with the answer key and determine if there really is a grading issue or not. And as always, you'll have a full week after posting of any score to notify us, and Will is the right person to notify if there's some mismatch between answers on the scantron and score in the gradebook or any such other troubling matters. Okay? Any other questions about the... all right, let's go ahead and delve into the eukaryotes then. So the next major section of the course, in fact, the next third of the course, will be walking through the different taxonomic groups. As we walk through each taxonomic group, we will consider what are some of the major important attributes of that group, how it is related to other groups, and then, because we don't have time to go into everything, pick a couple of particular key, interesting, biologically important phenomena that we'll highlight as we're discussing that group. And today, the group that is being highlighted are the protists. And the protists are shown sort of on figure 30-1 from your textbook, which is in front of you right now. By sort of, I mean that most of the names are not there. They're shown in orange, right? So everything basically in orange is a protist. And you can see as you eyeball the eukaryotic domain that, in fact, most of the branches are protists, but that the protists are clearly a paraphyletic group, meaning it's not a monophyletic group. It doesn't include all of the descendants. How do we know that? Well, look who it does not include. The protists do not include the fungi, does not include the animals, and does not include the land plants. These are three groups that share a most recent common ancestor with the other protists, but are not considered protists. This is clearly a paraphyletic group. It flunks the one SNP test for monophyly, for example. I also want to point out one thing about the conventions of this figure. Students often have a hard time going back and forth between trees when the branches are collapsed together as a single branch or more detail is shown. So, for example, in this particular figure up here at the top, we have the three domains, each of which is only shown as a single branch, and this shows the relationship between those three domains. Then in the part of the figure below, the bacteria and the archaea are still just shown as a single branch because we're not focusing on them, so we don't really care about the diversification between those lineages right now, this very second. And so we're just leaving those as undifferentiated single lineages. That makes sense. And so this says the same thing phylogenetically as this one. There's just more detail shown in this one for the eukaryotes. Okay, so you should feel very comfortable going back and forth between that. If it doesn't make sense to you, that's a great question for office hours. Okay, so protists then should not surprise you. We're an extremely diverse group. After all, look at all these lineages. In fact, your book identifies 7 major lineages. I'm going to switch colors here, so hopefully this will be less confusing. There's 1, there's 2, so there's a monophyletic lineage, there's a monophyletic lineage, there's another one, 3, here's another one that includes the land plants, there's 5 here, and 6, and 7. So there's 7 major different lineages, each of which has a long name with lots of syllables in it that's probably unfamiliar to you. And you are not responsible for learning the 7 different major lineages of protists or their distinguishing characteristics, okay? But certainly, as you're reading through it, I would encourage you to read about the diversification, and as you hit a name and if it's unfamiliar, then look to see, is this perhaps the group that we're in along with the other animals and the fungi, for example, and meaning we're in that overall group even though we're obviously not protists. Okay? It'll help you understand the relationships between these different lineages, although you certainly don't have to memorize all of these. Okay? So So if they're not a natural monophyletic group, why is it that they're grouped together with a single name, the Protists? Well, it's because it was early on, it was recognized that they were all eukaryotes and that they were sufficiently distinct from the fungi, the animals and the land plants, that they didn't really fall very neatly into any of those lineages. And so before phylogenetic trees like this existed, then it was natural to group them all. Protists are really important in a number of different ways. Your textbook has a very nice discussion of this, so I'm not going to go into detail on this during lecture time. I'm going to save that for a couple of other points, but I certainly would expect you to read this, the descriptions of how and why protists are important in ecosystems, and note that particularly aquatic ones, for example, protists typically live in a fluid medium, whether it's freshwater or marine, or even body fluids, meaning that they live inside another organism. And they are also important in global cycles, and so anything, of course, that produces CO2 or consumes CO2 is very important in a global sense if there are very large numbers, and protists are extremely abundant in many of the different ecosystems, and so even though a given protist may be very tiny, because of the sheer volume of the large numbers of protists, they have a large impact on global carbon cycles, for example. So please read about this. You have a good understanding of what that actually means. Many protists are also cause diseases in a variety of different taxa. In terms of the diseases caused by humans, the big major killer is in fact malaria. This is a really terrible disease. It's one of the biggest killers of all diseases afflicting humans. And the pathogen is in fact a protist. The disease-causing organism is a protist, and it has an extremely complicated life cycle, which we'll get to at a later point in time. But I'm going to go ahead and move on to, let's see. Sheesh. The next point about protists, which is their general lifestyle and body form. Most of the protists are pretty small. Most of them are only composed of a single cell, making them unicellular, but there are in fact a number of different groups which are multicellular or even colonial. Now, students often have a hard time differentiating between multicellular and colonial organisms, so I thought it would be helpful to show you a couple of examples. Let's go ahead and start with colonial example, which is actually here on the bottom as a little movie. This is a flagellate. I'll make it a teeny bit darker in here temporarily just so you can see this movie. This is an example of one of the species of a genus called Volvox. This is a fascinating group of protists that have been very well studied by scientists who are interested in the evolution of multicellularity because of different Volvox species show different attributes along the range of true coloniality versus getting towards multicellularity. So what is the difference here? A colonial organism is considered one in which the cells after division remain attached to each other. In this particular ball that's moving around is composed of a number of different individual flagellated photosynthetic cells which are attached to each other as a hollow ball. They have flagella, so it's capable of swimming all together. But the colonial forms are considered colonial as opposed to multicellular because the individual cells are basically identical to each other and you don't have differentiation. This is in contrast to something like the multicellular protist. An example here, these are example of brown algae. They're also called kelp, which may be a more familiar term to you. These form giant underwater forest equivalents in many of the shallow marine areas, including off the California coast. These are protists, and they're very large, and they are multicellular in the true sense, meaning that the cells from different parts of the body are distinct from each other. They have differentiated. So a cell in the holdfast that attaches the the kelp to a substrate is completely different in structure and characteristics than a cell, a photosynthetic cell up here higher on the frond, for example. So that's the difference between a multicellular and a colonial organism, is the extent to which the cells are differentiated from each other. That's the main thing. Okay, so what else can we say about protists? Well, protists can have sexual or asexual reproduction, or even both, in very complicated life cycles, and we'll see that later on, some examples of this. So they can have very, very complex life cycles. Considering general body type, one of the other distinguishing features of eukaryotes is that they're characterized by many different types of organelles. So this is very different from the prokaryotes, which have a much simpler cellular structure than the eukaryotes. And we're going to spend most of today then on the internal structure of the cells of eukaryotes, and particularly the organelles. So the eukaryotes have organelles that the prokaryotes don't. A fairly obvious example of this, of course, is the nucleus. Now there's an organelle that the eukaryotes have that the prokaryotes do not. And so there's interest, of course, in, well, how did this structure evolve? What are the steps involved in the evolution of this important structure? Well, this presumably happened quite some time ago, on the order of a couple of billion years, plus or minus. So obviously we can't view this directly, but there are many clues that one can use in the diversity of organisms that are still on the planet to put together steps that presumably occurred. So the steps that are thought to have occurred are shown here diagrammatically in figure 30-10 from your textbook, which is now shown in front of you. So if we consider an ancestor that did not have a nucleus, that's shown up here on the top, and so we've got the chromosomes. They're just there in the cell as a whole. They're not separated from the rest of the cytoplasm by any kind of membranous barrier. It is thought that the nuclear membrane evolved from elaboration of infoldings of the plasma membrane. So an intermediate step is shown here in the middle of this particular diagram, showing invaginations or infoldings of the outer plasma membrane. So this is the cell membrane that all cells have, including prokaryotes and eukaryotes. And one of the lines of evidence for such an intermediate step is that, in fact, some prokaryotes, in particular some bacteria, show various forms of elaboration of infoldings of plasma membrane, although never extensive enough to actually be comparable to what we see in nuclei. And then down at the bottom here, you can see that the elaborations have diversified to the point where the infoldings have actually broken off from or are now no longer continuous with the outer plasma membrane. We now have a complex membranous network here that completely surrounds the chromosomes and is continuous with the endoplasmic reticulum. And this is what you in fact see in all eukaryotes, is continuity, connection between the lipid membrane that separates the nucleus from the rest of the cytoplasm and also the endoplasmic reticulum as shown here. So this was thought that occurred in the evolution of the early eukaryotic cells, and it's certainly known that this has occurred very early on in the diversification of eukaryotes, because this is something that would be considered a synapomorphy for eukaryotes. It was shared by all eukaryotes, then it occurred before the diversification of the extant eukaryotes, meaning the ones that are still on the planet. Extant is a really inconvenient word because it sounds like extinct, but it means the opposite, so I'll try and enunciate very clearly, right? Extant eukaryotes, the ones still on the planet, not the extinct ones. Okay, so this then occurred presumably a very long time ago, and it is thought that the potential advantage of such separation is that we now have two compartments, right? We have the inside and we have the outside, And so, in fact, now transcription can be, and the processes associated with transcription and control of gene expression can now be completely separated from the processes of translation because they're separated from each other by a membranous boundary. If you can separate things into different compartments, you can have different conditions, and you can also have less crosstalk, less interference of components in one compartment with another if they're all safely sequestered from each other on other sides of a barrier. So this is you're going to see that this is a very common theme in eukaryotic cells. So let's now turn from the nucleus to considering some of the other organelles inside protists. Here's an example of a typical protist that some of you might be familiar with. This is an example of a ciliate. We tell it's covered by cilia. We've got a picture on the left. We've got a diagrammatic representation on the right. We've got some of the parts labeled. You can see that there's lots of different parts to it, lots of different structures. Some are very simple. Food vacuoles are example of very simple structures where food that's taken in is enclosed in a membrane and then broken down. Again, the separation of part of the cell from another part of a cell with a membranous barrier means that conditions can be different in one compartment than another because you have this barrier, you have this membranous barrier. So this allows for specialization, completely different conditions than inside the different compartments, which facilitates then a more elaborations of function that couldn't safely occur if they were not sequestered behind barriers. So these are all pretty simple organelles that we're looking at in this particular picture here, but there are some extremely complicated organelles, namely mitochondria and chloroplasts. Mitochondria are basically in virtually all eukaryotes with only a small number of exceptions, and chloroplasts, of course, are found in photosynthetic eukaryotes only, not in non-photosynthetic eukaryotes. But these are really complicated organelles, and so there was a lot of interest in how on earth did these more complicated organelles evolve compared to, say, something simple like a little food vacuole. Well, it is now appreciated that these very complicated organelles evolved within cells as a result of endosymbiotic events. This is referred to as the endosymbiotic theory for the evolution of some organelles. And I'm curious what percentage of you have actually heard of this before or have had some coverage of this. I see changes every year. The percentage of students who've actually learned about the endosymbiotic theory has been sort of gradually creeping up, but I'm interested in if you guys have been exposed to these ideas before. So please answer this clicker question. Have you ever heard of the endosymbiotic theory for the evolution of organelles before this specific class, right? Whether I'm mentioning it right now or are you reading it in the textbook? Yes, no, or perhaps you're not sure. Go ahead and click in, please. I'll just give you a couple of seconds. It shouldn't require too much time. Everybody clicked in? Super. Let's see. So about half of you have heard of it, which means, of course, a half of you have not. Okay, that's helpful. Thank you. Okay, so for those half of you who have, there is obviously going to be a little overlap with what you've learned before. And for those of you who are new to the idea, then let's walk through the steps. Let's first explain what these terms mean. So the name of the theory is the endosymbiotic theory or the endosymbiosis theory. What is symbiosis and what is endosymbiosis? We'll learn more about these terms such as symbiosis when we hit the ecological interactions between organisms part of the course. But for right now, here is the definition of symbiosis. The term symbiosis refers to any close and prolonged physical relationship between individuals of two different species. Okay, how does this differ from endosymbiosis? Well, in endosymbiosis, if we stick that prefix in front of the word symbiosis, then we mean it's so close and prolonged that one species is actually inside the other. One is inside the other. Okay, you can't get much closer than that, inside the other. Okay? And if you're talking about single-celled organisms, then inside the other means inside that cell, intracellular. So let's walk through the steps that would have to occur for if such a thing took place. So let's start up here at the top. We've got our ancestral pre-eukaryote over here on the left in blue, and then we've got some bacterium over here on the right. And if the organism over here on the left engulfed the bacteria, presumably this is feeding on the bacterium, but in fact, instead of breaking down the bacterium, it actually ended up surviving, then it is possible that such an association could develop, and this would be maintained, particularly if both members of this association got something out of this, if both benefited. Now, how would such a thing actually occur, and what is the evidence for this? Well, do the data support this? When these theories were first proposed, primarily by Lynn Margulis, several decades ago now, the scientific community thought this was the most ridiculous thing they had ever heard, extremely unlikely. How could it be possible that mitochondria, the powerhouse in all of our cells that is churning out ATP, on which all of our cells are so dependent, how could they possibly have any kind of bacterial origin? How nonsensical is this? What are the data, and what data would actually need to be provided in order to accept such a hypothesis? Well, some of the earliest evidence came from the feeding habits of single-celled organisms and eukaryotes. And so, for example, it's very common to see engulfment of food, such as shown in this static picture here from your textbook. This is figure 30-12, showing a organism that looks a bit like an amoeba, something engulfing food. Now, this is a static picture. It's a lot easier to see what's going on if we have a movie, so let's look at a movie. I'll make it a little darker so you can see the movie more easily. So here we are, we're looking at an amoeba. It's flowing around a food item right here. Whoops, okay, I just realized it's trapped, right? This is now completely encircled it. It's still moving around in there. Okay, it's on a loop. It's only about 17 seconds long, so you'll see it flow around. This particular kind of movement where the membrane-bound cytoplasm flows in one direction or another is a particular feature of one major lineage of protists. These structures are called pseudopodia. That's plural, as shown right here. And so this kind of engulfment then is seen frequently in many different kinds of amoeba or amoeba-like protists. But this is seen, the specific example we just looked at was an amoeba flowing around a food item, but all the other kinds of protists are also capable of ingesting and engulfing single-celled organisms like bacteria and so on. So this is not an argument that's specific to amoeba-like ancestors. This is just engulfing of food in general. So if then a bacterium is engulfed and instead of being broken down survives, then what would we expect the different partners to get out of this? And what would we expect the characteristics of such a bacterium to be? Well, we would expect the bacterium to share many features with mitochondria, right? So it would have to be an aerobic bacterium for one that was capable of using up oxygen, which tells you right away that this could not have occurred before there was sufficient oxygen in the atmosphere to have aerobic bacteria on the planet in the first place, right? Remember, thinking back, where did the oxygen in our atmosphere come from? Oxygen in the atmosphere is all of biological origin. You may or may not remember that it was the cyanobacteria that were the first oxygenic photosynthesizers. So the cyanobacteria, the very first photosynthesizers that were disgorging or producing large amounts of oxygen and filling, expelling such oxygen into the atmosphere would obviously take an extremely long time for oxygen levels to build up, but aerobic bacteria, such as the one that is the presumed ancestor for mitochondria, then would have to have formed after the formation of the oxygen, sufficient levels of oxygen in the atmosphere. Right, does that make sense? Right, okay, good. So we have to have an aerobic bacterium for one. And so the benefits of the association are shown diagrammatically down here in the bottom of the diagram, where this is what the endosymbiont benefits from getting oxygen, then and pyruvate then from the host cell. If it's engulfed, then anything entering the bacterium has to pass through or originate in the host cell. And what does the host get out of it? ATP. Okay? So, do the data support the endosymbiosis theory? Once it was discovered that there was actually DNA inside the mitochondria, then it was clear that there was one source of evidence that could be used to test the idea that mitochondria, in fact, were of bacterial origin, right? So I'll put mitochondria question mark up here. Because, of course, scientists who felt that it was unlikely that the mitochondria were of bacterial origin could simply have an alternative hypothesis that the DNA inside the mitochondria was just simply pinched off somehow from one of the chromosomes or part of the genetic material of the eukaryotic host. It didn't necessarily have to come from bacterial origin. Once the mitochondrial DNA was actually sequenced, then it could be compared with the DNA within various eukaryotes and also bacteria to just try and figure out, well, what taxonomic group does the mitochondrial DNA actually most closely is most closely related to? And it clearly came out as a sister group to one particular group of bacteria. So if one takes mitochondria, the organelles, the DNA within the mitochondria, and puts that on a phylogenetic tree by comparing similarity of the mitochondrial DNA with the DNA from these other groups, then it does not come out with any of the eukaryotic groups, but rather as a sister group to the alpha proteobacteria. So therefore, it is assumed that this, move this out of the way, this bacterium was presumably an alpha proteobacterium, or alpha proteobacterium type bacteria. Right? Does that make sense? Does that make sense why that's evidence for the endosymbiotic theory? Excellent. Of course, that's not the only source. Here's a whole long list here that I made for your convenience here. So if we look at mitochondria, well, they're about the same size as bacteria. They also replicate by fission, just as bacteria do. And this duplication is independent of the cell division of the host. And they have their own ribosomes that are much more like bacterial ribosomes than they are like eukaryotic ribosomes. There are distinct differences in the enzymes and some of the structural proteins of ribosomes. And mitochondria, as mentioned, has their own genome, but in addition to being more similar in sequence to the alpha proteobacteria than any of the eukaryotes, it's also circular like bacterial chromosomes. And there's also a double membrane seen in the mitochondria. What does the double membrane, what's the significance of that? If we go back to this diagram and see where a double membrane would come from, if you consider that a bacterium already has its own membrane, I just colored it in green, when it's engulfed then and taken into the cell would fold around it and pinch off. So we now have two different layers of membrane, one within the other. And so it's thought that this double membrane is also evidence of a bacterial, early bacterial endosymbiont then being taken into the cell and developing a double membrane. Okay, so that's the story for mitochondria, very quickly summarizing many, many decades of work by a very large number of individuals. How about chloroplasts? That's another very complex organelle that is thought to have an endosymbiotic origin. So there is not a comparable figure to figure 30-8 for chloroplasts. I went ahead and put it up in front of you. This is the one that shows the acquisition of an alpha proteobacterium-like bacterium in the evolutionary history of mitochondria from a bacterial precursor. But if we consider now what the steps were like for chloroplasts, many of the details would be somewhat similar, in that it is thought that a bacterium was taken in internally, but of course now the details would be different, right? Instead of an alpha proteobacterium-like organism, we would expect something that is photosynthetic. A photosynthetic bacterium that would be taken in. And now, of course, the benefits would be quite different for the host and the endosymbiont. Now what the host gets out of it, so this is now the chloroplast version of this particular figure, the host would get oxygen, right? Because that's a byproduct of photosynthesis, gets oxygen and also sugar, like glucose. And what would the endosymbiont get out of it? Well, just like the arguments made for the other endosymbiotic bacteria, would get protection. If it's a photosynthetic organism, it would also need access to light, so this would be something else that the host would have to provide, is at least occasional access to light. And then, of course, any nutrients that the photosynthetic bacterium would need would have to be provided by the host cell, because after all, it's engulfed in the host cell. So that's what the photosynthetic bacterium would look like. Now, we went through the list of the various forms of evidence for bacterial origin for mitochondria. Let's now do the same thing for chloroplasts. This will actually be really quick and easy because it's very similar. There's a list for chloroplasts. I have just contrasted it with the mitochondrial one. Chloroplasts are also about the same size as bacteria and replicate by fission and have duplication independent of cell division and have their own ribosomes and have a circular genome. And the DNA sequence is now most closely related to familiar cyanobacteria, right? The first bacteria that had oxygenic photosynthesis. So it is thought that if we go back to this picture then, specifically, this would be then considered to be a cyanobacterium of some sort. Okay. The scientists were looking at the evolution of chloroplasts in the eukaryotes. They noticed a curious phenomenon. This is figure 30-14 in front of you right now. And so in trying to figure out where in the tree, the phylogenetic tree of the eukaryotes, did this presumed origin of chloroplasts take place, is thought to have occurred right here in this particular location, in this particular branch. And so this would be then the incorporation of cyanobacterium as a sort of pre-chloroplast. Right? So all of these descendants then in this monophyletic group share many characteristics of the chloroplasts. But in studying the photosynthetic ability of some of the other protists that are also shown in this particular part of the phylogenetic tree of the eukaryotes, it was noticed that there was another group shown down here at the bottom that also had chloroplasts, and yet these specific chloroplasts were considered very similar to, so the chloroplasts in these groups were considered very similar to red algal chloroplasts, which seemed a bit odd because after all, this group here is not considered a sister group to the red algae, and so how on earth could this occur? And in addition, there are a couple of other groups, this one and this one, in which the chloroplasts of these particular groups are similar to the to the chloroplasts in green algae, so green algal chloroplasts. Okay, so what's going on here? There's a couple of different possibilities. One is that the phylogenetic tree is in fact incorrect, and that this group down here is actually more closely related to the red algae and shouldn't be over there off on its lonesome. This is not considered to be the true answer to this question because, after all, it is just the chloroplasts specifically that are comparable. Everything else about the rest of the body, this is only one organelle, everything about the rest of the body of the ciliates and the dinoflagellates and so on is in fact extremely distinct. It's just the chloroplasts. And this is why it is thought that in order to understand why these different groups have chloroplasts that resemble red algal or green algal groups is because of secondary acquisition, meaning that these groups here, the euglenids, and this other group here in the rhizaria, then the chloroplasts resemble the green algae because members in these groups swallowed some green algae, and those were then the precursors for the chloroplasts in these particular groups, right? So that means instead of just acquiring chloroplasts from swallowing cyanobacteria, you can swallow a descendant of something that had swallowed cyanobacteria, right? Does that make sense? So what lines of evidence are there for that kind of secondary endosymbiosis? Well, there are numerous examples of photosynthetic protists that live inside the cells of other organisms. I don't think this is from your textbook. This is just a gorgeous picture of some stunningly attractive protists that are chock full of another protist, right? So these little green circles are, each one of those individuals is a green alga, and they're filling up the insides of the cell of this other protist. So protists do live inside other protists, as shown here. In fact, not only do protists live inside other protist cells, protists also live inside cells of other organisms, even some animals. And so, for example, corals, which is a kind of animal that we'll be discussing later, in their tissues then, they host photosynthetic protists of a couple of different protist groups. And this is a very close symbiotic association. So there are many, many examples. This is just two. There are quite a large number of associations then where protists do inhabit the cells of other organisms. So it's not that far-fetched then. Other evidence comes from the number of membranes that are around some of the chloroplasts in these other groups. This is the logical steps are shown here in figure 30-13 from your textbook, which is shown in front of you right now. And so what this shows is if a protist, shown over here on the left, which is at the current time not photosynthetic, swallows a photosynthetic protist, which is shown here in orange, which because it's photosynthetic, it already has a chloroplast, right? Then over time, if changes given enough time, then changes could take place in this endosymbiont, and it can then, over long periods of time, then basically turn into an organelle from its previous protistan independent life. And under those circumstances, then one would expect a larger number of membranes, just as we showed earlier, where the original acquisition of a single bacterium would lead to a double membrane. If you then swallow something where the chloroplast already has two membranes, and there's a third, and then there's a fourth. So you'd expect all the membranes to sort of line up and have even more of them. Let's see. Let me go back here actually for a second before I go there. Yes. Before I leave this particular point, I want to make a really important point here, is that as things are going on in the different lineages, it can be very tricky to figure out what's happening when, and that's what the clicker question is going to be about in just a couple of seconds. But I want to introduce the idea first. I think that'll make it easier for you. As mentioned before, in a phylogenetic tree like this, time, of course, is going from left to right, meaning that more recent, in this particular orientation, more recent is shown to the right, earlier is shown to the left. But when there is a representation like this without a time scale on it, you cannot assume that everything is lined up in perfect register according to time. That is, the information in here is showing what happened before what, but they're not perfectly lined up. As an example of what I mean by this, let me grab another color here to make this point more clear, because I think this is an important one. Notice this event right here that I circled in red. This is the presumed point of acquisition of a red algal endosymbiont. Okay? That presumes that the red algae as a lineage have already diversified from the other groups. Okay? So if we then just look up here, notice that red algae are not even diversifying until a later point in time. Okay, that's what I mean by you can't assume that everything is lined up in perfect registration on something like this. Okay? Does that make sense? Yes, no, maybe? Well, one way of evaluating that is to give you a clicker question where we can consider changes in time. Let's consider this one. Any response counts. This is an example of a test question from a previous year. So, consider a lineage of bacteria. This is a real lineage of bacteria, the Chlamydiales. They are all obligate endosymbionts. What does that mean? Obligate, meaning they're obliged to. They have to. This is the only place they live. They live inside animal cells, right? Endosymbionts inside animal cells. That's the only place they live. They do not live in any other kind of eukaryotic cells. Just animal cells, specialized. When do you think they evolved their metabolic strategy? You can answer the question by filling out the end here with one of these four choices. They probably evolved their metabolic strategy as an evolutionary innovation before the split between the bacteria and the eukaryote lineages, before the split between the archaeans and the eukaryotes, before the split between the fungal and the animal lineages, or after the split between the fungal and animal lineages. Why don't you take 30 seconds or so to chat with your neighbor and think about this, and I'll check back in with you. Okay, has everybody formed an opinion, clicked in? Let's see what you guys think after this stabilizes. Okay, looks like it's stopped. Let's see what you guys think there. Wow, all right. 44% of you got it right. Congratulations. Unfortunately, that means more than half of you missed it. But what's the way of thinking about this? Well, the animal cells, this is the environment for these bacteria. Okay, so the environment has to exist before they can evolve to specialize in it. Okay, so that's why it would be logical to assume that this is after the animals split off from the other lineages, such as the fungal lineage, okay? So certainly not before that. This is similar, by the way, to what we were considering if this is, if the chloroplasts within this big group here are thought to be derived from a red algal precursor. Hang on, shh, just a second. if the chloroplast in this particular group is assumed to be more closely related to the red algae than, say, the green algae or any of these other groups, then this means that the red algae, this event could only have occurred after this lineage of red algae diversified from the other groups and specialized, accumulating differences within just that one lineage that then distinguish them from the other lineages. So the next topic is complex life cycles. So we're going to go ahead and hold off until next time, and we'll start with complex life cycles. I'll see you then.